So here's where we're going to get into the details of each of the sections of interphase. The first section is the G1 phase, the growth phase. It's called gap 1. We often refer to it too, though, as the growth 1 phase. Pretty much the cell's growing. It's got to double in size because if you think about it, it's going to split into 2 new cells. And so in order for it to be able to do that, it has to be big enough to make that split. You don't want to have one cell that then splits into 2 and they're these tiny little cells and then they can't function properly because they're not big enough. So what ends up happening here is this is the phase when it's time for all the cells to grow and copy, all parts. That includes like your organelles, your mitochondria, vacuoles. We literally double everything. That word double is really important because the cell will need to double in size. That just makes sense. If I'm going to take the cell and split it into 2, I want it to be double the size. That way when I split it, it's back to normal. There's a checkpoint, a special protein that comes in called RAS cyclin. This protein will check for that size regulation. If it's not big enough, it's going to send the cell back to do some more growing, or your new word of the day is apoptosis. The cell will die. It will destroy itself thanks to some of our immune cells, which will come in and help the cell just give up on itself. And so we call cell death apoptosis. Sometimes the cell just can't get itself together, and instead of it leading to more problems, the cell will just destroy itself. In most cases, the cell does everything it's supposed to. All errors, or it is error-free, I should say, and there's no issues. So the cell will just continue from the G1 phase into the next phase, which is our S phase. This goes back to what we did when we covered DNA replication. We're going to actually copy the DNA. Before we copy the DNA, though, you'll see that there's a checkpoint that occurs. We have to go in, or the cell goes in, I should say, and checks itself to make sure that the DNA is healthy enough to be copied. We don't want to copy unhealthy DNA because then, of course, that unhealthy DNA is spread everywhere, and that can lead to problems. So right here, you would put DNA. That is where the copying of the cell's DNA takes place. Once the checkpoint has been cleared, DNA has been copied, just like we did when we did DNA replication, and you'll see that in the next slide. Keep in mind that that strand gets unzipped, that DNA strand, and one piece goes this way, one piece goes that way, and then we make our complementary strands off of that. That means that one piece of the strand is old, and then you make a new copy. So we call that semi-conservative. And that's important for you to know because, let me stretch this out. We don't want DNA that's too old. You need that DNA to sort of have some of what it was before, but then also be new, because over time that would potentially cause problems if the DNA were constantly being copied, but all the old strands were staying together. That means it could lead to some more problems. So it's actually quite beneficial that part of our DNA strand is new and part of our DNA strand is old. And that's what we mean by semi-conservative. Here is another example, and we did this already, so this should be pretty familiar to you. If you notice here, you have your original strand and it gets split. Each strand is then base paired all over again. And what you should notice is that this ATGC, ATCG should match over here, ATGC, ATCG, and the same here. You should end up with exact copies. That's the whole point of DNA replication. And then, of course, finally, if we look at our last phase, or I should go back for a second. Let me go back to this slide just to remind you, there is a checkpoint that's at the end of the S phase. We have to go back and check and make sure that we didn't have any copying errors, in the same way that I went and checked your work when you were doing DNA replication to make sure that you base paired correctly. there's a checkpoint here to make sure that the DNA has been copied correctly so we don't have any mutations or any other kind of errors. So going back to this slide now, we're finally ready for mitosis. We're going to continue growing and preparing. We're going to make sure we put together all the other pieces of the puzzle that need to be in place for mitosis to take place. So the cell here will continue to grow. The cell also starts organizing itself to be able to prepare for mitosis. And mitosis, of course, is the actual cell division that's going to take place. One thing you do have to be familiar with is there is a very special organelle that forms only during this time. That's called spindle fibers. And we're going to talk about these with our simulation. Those spindle fibers are really important. Imagine them kind of like a fishing line. And the way that you go fishing and you reel something in, the spindle fibers actually come in and they grab the chromosomes and pull them apart to each end of the cell, so that way the cell can separate the chromosomes properly. Those spindle fibers are critical to cell division, and so we do talk about those in detail because they're a special organelle that doesn't exist at any other time except during this cell division process. You can take a look at your independent practice now, and you can, of course, revisit this information about interphase. Tomorrow, we do have PSAT testing, so I will not be pushing out a lesson, but we will do a lab simulation on interphase, and I will have it set up to where those of you who are at home will still be able to see the lab and participate in the work that goes along with the lab. So that'll be coming up on Thursday.